Welcome to today's workshop on image segmentation and 3D analysis. My name is Ronell Sicat from the KAUST Visualization Core Lab. And we've been doing this image segmentation workshop for the past few years. But this time we're going to focus on AI assisted tools, which is kind of the trend, as you all know. But I'll still go through some of the basic stuff because we need those like basic knowledge to use Avizo and Ilastik, for example, before we can use the AI assisted tools. So just a quick note, the workshop will be recorded and we're going to post the workshop on YouTube, but please still stay. So did you guys, or were you guys able to install Avizo and Ilastik? Yeah? Okay, cool. So we're going to do a bunch of hands-on stuff. And I'm also providing some PDFs and videos where you can follow for the AI tools for later. So just some quick introduction of the Visualization Core Lab before I get into the workshop materials. So the KAUST Visualization Lab, or KVL, provides support and collaboration in data science and data visualization. So we have different fields of expertise, such as scientific visualization, information visualization, data science, and machine learning. And for today, image segmentation and 3D analysis. So if you need help in any of these fields, feel free to reach out to us. I'll show our email address and website in a little bit. So aside from expertise, we also provide facilities, right? So you've probably seen the big walls when you pass by Building 1 along the spine. So we have big 2D display walls and VR displays as well. And if you want to know more about the Visualization Lab, you can visit our website or you can send us an email. If you have questions about the lab or if you need help with visualizing or segmenting or analyzing your data. Okay? All right, so for today, this is sort of our schedule. I'll give a quick workflow overview. So just an overview of the steps that we will be doing today, and I'll also give a preview of the types of data sets that we'll be working with today. And as I said, first I'm gonna give an introduction of basic segmentation tools in Avizo as a foundation to help us with using the AI assisted segmentation tools in Avizo. Then we're gonna have a break, and then we're gonna switch to the segmentation tools using Ilastik. And finally, so after we've done the segmentation, most likely you want to do analysis on your data, right? So you want to do some measurements. So I'm going to show some common analysis workflows. So how to get some measurements or analysis from your segmentation results, all right? So let's take a look at the workflow overview. So hopefully you are here today because you somehow acquire your image stack data. So it's 3D data, right? But it's a stack of 2D images acquired through some sort of imaging modality like microscopy or electron microscopy, confocal microscopy, CT scans, etc. And we're interested in performing segmentation, right? Which is basically separating, so classifying and separating the different materials in your data. So for example, here, this data set is a sand-packed material. So you have sand particles in bright gray and the air or void in black or dark gray. And we want to separate these materials, right? So that later on we can do some sort of analysis, like, for example, counting the number of sand particles or measuring the fraction between sand and air. Right? And I mean, the applications are, let's say, there's a wide variety of applications, right? So this is just one example. You could be working with biological data, some rock samples, some membranes, etc., right? But you somehow would have a similar workflow. So you have your data, you do segmentation to separate the materials, and then run some analysis. So this is what we'll be focusing on today. So just some important concepts, right? So if you look at your data, if we look at a tiny piece of it, it's basically just a bunch of numbers, right? this is what you acquire from your imaging devices or imaging modalities. It's just a bunch of numbers. And to help us make more sense of these numbers, we apply what's called a color map or transfer function, right? Where we basically apply color and opacity or transparency to these numerical values so that you see something more meaningful, right? And if I want to, I can change the color map, right? So that would change the appearance of my image, but the underlying numbers are the same, okay? And we sometimes do this, like we change the color map to hide some things in the data or to emphasize some parts of the data, okay? And our goal in segmentation is to create a new, let's say, a new type of data, which consists of, in our case, just integers, right? So numbers from 1, 2, 3, 4, 5, and so on, where each number corresponds to a material, right? So in this case, let's say for the air pixels or voxels, we assign a value of 1, and for the sand particles, we assign a value of 2, okay? Makes sense? And 0 usually is assigned to like the default label, which is unlabeled or unassigned pixels or voxels, okay? And obviously you can do this segmentation manually, right? You can go through pixel by pixel and decide, is this air particle or sand particle? But of course, we don't want to do that because that's tedious. It's gonna take forever. That's why we have segmentation tools, which we will talk about today, okay? And finally, after you have your segmentation, then you can do some more interesting things, right? Like analysis. So for example, you want to count the number of air particles and sand particles. So in this very simple example, we get 7 and 9. Of course, I mean, this is just a small example. If your data set is big, you don't want to do this by hand. Or you can get the fraction, for example, like that says air is 44% and sand is 56%, and so on. And this is just a simple example. You can have more complex analysis that I'm gonna show in a little bit, okay? But this is what we will be working on today, segmentation and analysis. So now I want to do like a quick preview of the data sets that we will be looking at today. So you've already seen this, and I like using this. It's a simple, small data set that's built in to Avizo. It's the sand data set. And I like using a simple data set so that we can focus more on the process, okay? By the way, if you want to do the hands-on stuff, make sure to download the data sets, okay? You'll find it in the training page. On the left panel, I can quickly show you. So this is the training page. Over here, there's a link to download the data sets, okay? All right. All right, so the next data I'm gonna show is this electron tomography of a zeolite. So it's a very small material, let's say, and I'm gonna show you in less than 5 minutes how we can extract majority of the macropore regions, right? So it's these like small spherical shapes, okay? And yeah, in less than 5 minutes, we can get a nice, not perfect, but nice segmentation of the macropore network. And then you will be working with this cryo-EM of a cell. So it's up to you. You can pick which part of the cell you want to segment. So you're gonna do this on your own later. And then we're gonna use Ilastik to segment the mitochondria and the boundaries of this cell EM. And we're going to take a look at this example of a confocal microscopy of a cell, how to extract these tubular structures using Ilastik, okay? And then in the analysis part, we're going to take a look at a CT scan of this coral, and we're going to do some measurements, like measuring the pores inside versus the more dense material, measuring lengths. So some of you indicated in the registration survey that they want to measure lengths and angles. So we're going to take a look at that. And finally, we're going to take a look at core plug CT. So this is more for people from Antwerp who does this a lot. we're going to segment the pores and quantify them. And what you see here on the left is a more complex type of analysis. It's more of a simulation that Avizo can also do, but we're not going to really do that today. But just to let you know that it's there. All right. Cool. So that was our overview, and now we're going to start with basic segmentation using Avizo. All right. All right. So we're going to use Avizo 2023.1.1. Okay. So this is the latest version, and you can find it in the KAUST IT website. I provide the link on the training webpage. Now, the AI tools for segmentation have some requirements, okay? So unfortunately, it's only supported on Windows OS at the moment. You need an NVIDIA GPU, okay, to facilitate the computation, and you need to install the deep learning environment, okay? And again, the instructions are there on the training website on how to do this, okay? But hopefully you've done this before, right? Okay, and in the registration survey, I also asked for the participants' experience with Avizo and Ilastik, and majority of the people don't really have Ilastik and Avizo background. So for the first example, I'm just going to do a quick demo, okay? So just watch and take a look. Try to have a big picture idea of how Avizo works. And yeah, this should help you with the next hands-on steps, okay? So let me start Avizo. Okay. So again, for this, just have a look, okay, just to give you an idea. So for you guys in the back, the text might be a bit small, so you might want to move closer. The first 3 rows are the best. Can you see the text? It's okay? All right, cool. So this is Avizo. When you open it, it looks like this. And we're going to open a data set, so just click this Open Data button. So this is the datasets folder that I shared with you. There's a lot of stuff in there. And I'm just going to start with the SAND dataset. So open the folder. And here you can already see that my dataset consists of a bunch of 2D images, right? So this is my stack. So I just do Control-A and open. And then you'll get a prompt to specify the name of your object, the units. So this is very important if you want to do measurements and analysis with units. So you'll have to specify this, and you should know this, right? You're the one who acquired your data. You should know the units. So in this case, I'll just say my units are in microns, okay? All right, so after that, my data gets loaded right away. And if you look here on the upper left, this is your what's called project window, right? And this is where your data objects are in. So this green icon here represents my data, and Avizo automatically attaches a visualization module to it, which is an orthoslice. And the visualizations show up here on the right window. So this is a 3D visualization window. So, and if I click on the orthoslice module, you're going to see here on the lower left the properties, right? So whatever object you click here in the project window, the properties show up on the lower left, okay? And for example, the orthoslice, I have this slice number that I can change and to indicate the slice number that I'm interested in, okay? All right, and yeah, so usually when we start with a new data set, we first want to see what the data set looks like, right, before even segmenting it. We want to find out more about the data. And the first thing that I would usually do is attach an orthoslice and have a look. But even better, so we can attach a volume rendering module to it, which is a 3D visualization. And to do that, so we can click this white circular part of the icon here, and then you're going to see some of the most commonly used modules here. And I'm just going to click volume rendering. You can also type the name of the module here. yeah, if you're searching for modules. I'm just going to click that, and then you see here volume rendering. So let me just turn off the ortho slice. So you can click this blue button here to turn it on and off. And now all we see is the volume rendering, which is a 3D visualization, right? And with this, we already have a kind of just a basic picture of how the data looks like, right? And we see the particles already. So I won't go into the details of volume rendering. So this is more, hopefully you attended the basic Avizo workshop. But if you want to know more, you can reach out to us, okay? So anyway, we're gonna go ahead now and try to segment this, right? So I want to extract the sand particles, all right? So let me turn this off. And what I'm gonna use yes? Yeah. Yeah. So the question, for the sake of the recording, is that is there a way to just show, instead of like this, let's say the smooth volume rendering, just cubes? This is actually called voxelized rendering. Yeah, we can do it. Actually, I'm going to show it because you can apply the voxelized rendering on the segmentation data. Because segmentation data is not really binary, but you can think of each voxel as an opaque material. I mean, let me show it, okay? Let me do that. Yeah, exactly. So I didn't want to say binary because once you have more than one material, it's not binary anymore. I mean, yeah, exactly. And that's what we're going to have for the beginning, right? So let me just go ahead and do that, right? And to do the segmentation, okay, so I'm not using any AI tools yet, right? As I said, I want to show the basic stuff first. And in this particular example, I'm going to use interactive thresholding, right? So once I'm not sure yet if it's visible, it says interactive thresholding. So what this allows me to do is specify a threshold value that I'm changing right now. It's basically a threshold window, right? So all that threshold window is saying is that any pixel or voxel with a value between, let's say, in this case, 7,451 and 53,000, separate that, right? Like put that into my segmentation. And you see here on the right side a preview of which pixels would be included in that threshold. And I mean, I can actually do this. I can click apply and turn off the thresholding module. And this new module that's been created is actually the segmentation result, right? And now let me show you, I mean, just quickly the voxelized rendering. If you click on that white part there, already it's going to show here the voxelized rendering option. And this is it. And if you look, let's create it. So you're going to see here, you basically have these cubes, right? And I hope it's clear with you, but basically each voxel is rendered as a cube, okay? And this makes more sense for segmentation data, all right? Does that answer your question? Yeah? Yeah, no, it's better to, yeah, exactly. And one thing you will notice is that the output is noisy, right? So you have these tiny particles in between that get segmented. And in order to fix that, we're gonna actually do a bit of denoising, right? And I'm not gonna go through the details of this, but actually if you look at the thresholding preview, you're gonna see already, you can anticipate that there's gonna be noise over here, right? So what we can do is apply a filter. Again, I'm just gonna go through this quickly because it's not really the main topic for today. So I'm adding a median filter. So there's other filters that you can explore. I'll mention some of them towards the end of the workshop. I'm gonna click apply. And then just to show you the difference, if I apply So now I basically changed the input to the thresholding to the cleaned up version of the data. And as you can see, you don't have the salt and pepper noise anymore, right? So now when you apply the thresholding, you should get a much cleaner segmentation, as you can see here, okay? So yeah, this is kind of a very simple example, right? So you have your data, which is usually noisy, then you apply some filter to denoise, and then once it's denoised, you can do segmentation. And in this case, I used a very simple method, which is thresholding. And actually, personally, I try thresholding first all the time, right? Because it's a super simple method, super cheap to compute, and sometimes it works, right? As you can see in this example, it's enough to do the job. All right. Okay, and then now that I have my sand separated from the air, I'm going to show you also quickly what then you can do, right? So let's say I want to count the number of sand particles, right? So what I want to do is next separate the individual particles from each other. And in Avizo, there's a module called separate objects, right? So actually, if you click on this white icon here, as I said, and just type, you're going to find the module. Click that and apply. All right. So it generates a new version of the data set, but now the particles are separated. So I can take a look, and now you see here, for example, that there's a crack that separates these two particles. And even better, we can do a label analysis, which will assign a different material to each particle, okay? And you get something like this. All right, so it separates the particles, and at the same time, it does some measurements for you, like volume, surface area, and so on, okay? All right, so in this example, we actually did the whole pipeline, right? From looking at your data, segmenting it, and doing some simple analysis. And again, I just wanted to show you this, because this is kind of the main workflow. And something, like an important thing to point out is that we did all of this in the project window, right, over here, just manipulating stuff here. So I mentioned that because in the next example, we're going to use the segmentation workroom, okay? And that's where the AI segmentation tools will be, okay? It's not going to be here, all right? But yeah, this was more to give some people who are not familiar with Avizo just a general feeling of how Avizo works, okay? Yeah?
Just a short question, is there any form of segmentation?
Yeah, well, you're the expert, so you have to inspect, right? So that's a good question. Yeah, your eye. Well, it depends. So for example, I mean, let me talk about the first option first, which is by eye. So you're the expert in your data, so you should be able to judge if your segmentation is correct. And one way of doing that, or a common way of doing that is, so you have the ortho slice that shows your original data. And then what you can do is on the ortho slice, attach a color wash module. And then the color wash, attach it to your segmentation. So let me use the colored one. So this will allow you to visually inspect, right? So you can go through the slices and just see, well, okay, this looks correct. If not, you can either tweak your segmentation parameters, or worst case scenario, you do manual segmentation, right? And since you mentioned that, it's very important to know that after doing the segmentation process, most likely you will not get a perfect result, right? So you'll probably have to do some fixing, which is sometimes called proofreading, right? So you'll have to do maybe a little bit of manual work. But the goal of using segmentation tools, especially the AI tools, is to reduce the manual work, right? And then the other thing is, like, for example, I have a student who did a scan of a material that he 3D printed, right? And he knows the, let's say, the volume of the 3D print material versus air. So you can also have some sort of quantitative check, right? So you can measure the segmentation result and compare it with your experimental data, okay? And yeah, it depends on the situation. But the first and easiest is just visual inspection, okay? All right. Everything okay so far, I hope? So I'm gonna close this. And now we're gonna go into the segmentation workroom, okay? As I said, that first example that I showed is, it works, right? It works, and you can use it, and you can use like different modules to do segmentation. But for the AI segmentation tool, we'll have to use what's called the segmentation workroom. And that's what I will show you now. And if you want, you can also follow along. If you have Avizo with a deep learning environment working, okay? So I'm gonna start from scratch again. I'm gonna start Avizo. Let's open the data set. Again, that's just this open data button. Go to the datasets folder that I shared. Go to the SAND folder, Control A, open. You can specify the units. All right. And yeah, so for this data set, now I click the data set, and then you're gonna see there's this segmentation button on the top. So if we click that, usually you get a prompt like this in the beginning that will ask you, do you want to use the old classic segmentation workroom or the new segmentation workroom? So for us, we want to use the new segmentation workroom because that's the one that has the AI assisted segmentation tool. Okay, so we're gonna click OK, or we're gonna click that. And this is how it looks like, right? It looks a bit confusing in the beginning, but it's actually simpler than the classic workroom, okay? So anyway, let me go through the parts. So on the upper left here, we can specify the image that we want to segment. So if you go to this drop down, you can click this one, and then it loads it to the slice views. And here, the label image, this is actually the output segmentation, right? And because we don't have anything, we want to create a new label. And then the patch set, we won't use this until we're gonna do the AI segmentation. So for now, we're okay with this. And then over here is the list of materials, right? So we have the default exterior, which is assigned to number 0. Material, you can double click it and then change it to, let's say, sand. You can click this one, create new material, to create another one, let's say air. And then obviously on the right side you have the slice views. And also here you can use the brush. So if you look here on the top, there's a brush and eraser. So if you click the brush, you can brush on the slices like this. And you can press F to fill the region. And what I did here was basically just do a selection, right? So it's just highlighting some pixels. They're not assigned to any material yet. And now if I want to say, okay, these selection or these highlights, add it to sand. So I click sand over here and then click add, all right? And once you do that, you're gonna see it changes color to green, which is the color of assigned to the sand material. And for example, I want to add some materials to air. I'm gonna do this. So this is obviously the manual process, right? And you don't wanna do this. I'm just showing you the process. So I click air and then click add material, okay? And yeah, basically we've done our first segmentation, right? Using the segmentation workroom. And you have a 3D preview here as well. So if you click over here, you can preview your segmentation on the 3D view. Okay, for some reason it doesn't, okay. If you want to rotate, press and hold Alt, and then left mouse. So it gives you a preview of your segmentation in 3D, okay? All right, so as I said, we don't want to do manual segmentation, right? And that's why on the lower left, you have your different segmentation tools, right? And you have here thresholding, which is exactly the same as the thresholding tool we had before. We have other things like watershed, texture, classification, and the AI assisted segmentation, which we will focus on in a little bit, right? But for now, I just want to show the simple one, which is thresholding. So once you click thresholding, I'm not sure if you see it, but this is your threshold window, right? So you set the minimum and maximum, and then you see the preview on the left, which is in light blue. So you can change the threshold parameter over here. And let's say you're happy with this threshold, you click this create selection, and you get a preview on top. As I said, this is just highlighting, it's selecting, it doesn't add it to the material yet. To add it to the material, you have to click the material and click add, okay? And now it's sent, all right? So this is very important because we're gonna use this in the AI segmentation tools next, all right? Okay, so I will not go through the other tools because if you look at we've gone through them before in past image segmentation workshops. And if you're interested in taking a look, you can just search in YouTube for KAUST Visualization Core Lab. We have the recordings of those workshops from before, okay? All right, before I move on to the next example, so right now, as I said, we're in the segmentation workroom, right? And if you now click on the project over here, it brings you back to the original project view. And you see here, there's a new object that's been created, and that's actually the segmentation that we did. So again, if we look at that together with the slice view, this is how it looks like, right? And we can also, again, see it in 3D, okay? And that was the result that we generated using the segmentation workroom. All right. Any questions so far? All right. Very easy, right? I mean, okay. All right, so that was kind of a super quick review of the basics of segmentation without the AI assisted tools. So the most important part there is just getting familiarity with the segmentation workroom, because it's the same workroom that we're going to use for the AI assisted segmentation. All right. Now, before I go into the Avizo tutorial, I just want to share one slide about some important concepts, right? So for today's workshop in Avizo and Ilastik, we're going to be focusing on using deep learning models for segmentation, right? And most of you would be familiar with deep learning, and the idea applied to our segmentation task is you have your raw data, right? Your raw grayscale data, and you want to segment some parts of it, right? So in this example, there's different parts of the cell that you might want to segment, like mitochondria, ribosomes, these tubular structures, and so on. And obviously, in the case of data sets that look like this, it's not easy, right? So if you try to use the thresholding tool on this one, you're not going to get anything meaningful, right? And yeah, it's not an easy task. But visually, you might see that for some structures, like the ribosomes that are circular, there's a pattern, right? For us humans, it's kind of easy to recognize. And for example, the tubular structures, obviously, that's also visually clear to see. And for things like this, it's very helpful to try to use deep learning models to try and learn those and segment those automatically, right? And yeah, I mean, it's not an actual robot, but it's an algorithm, right? And the idea is that you have your raw data, and you as a human will provide some examples, right? Like the brush strokes that you see here. And this is kind of the ground truth, or let's say training data, right? It's like teaching a child that doesn't know what cats are and showing them pictures of cats or showing them actual cats so that in the future they will recognize cats. The same here for training your deep learning model, you have to give it examples, right? Like, hey, model, this is how these tubular structures look like, okay, in my data. And in general, the more data you give for training, the better. But of course, the more time you have to spend, right? Because this is manual work. And the idea is that after training, your model learns about how to separate this particular material or class from your images, and it will do most of the job for you, right? Again, most likely it's not going to be perfect, but it will do a lot of the job, right? So it could reduce work from, I don't know, 1 week to 1 day, yeah? But this is the idea, okay? And for today's workshop, we're only going to tackle the AI tools in Avizo and Ilastik where the model is given to us, okay? So we're not going to deal with like engineering the models. Like we're not going to decide how many layers there will be, what type of layers, and so on. Although in Avizo, to some extent you can do that, but for today's workshop, we're just going to focus on the tools that give us the models already, okay? Yeah, I'll talk more about that later. But this is the general idea, okay? You have your data, you give some examples, and you ask the model to do the segmentation for you. And you can iterate the process, right? If you're not happy with the results, add more training or tweak some of the parameters, right? So before we get into the hands-on, again, I want to show one quick example of how it works, okay? Again, just have a look, get a big picture idea of the process so that later on you can do it on your own. And again, I will do it on this zeolite data set. Unfortunately, I cannot share the data. It's just been published, by the way, in Nature. But I want to show you how quickly it is. Sorry, not Nature, Applied Chemistry. I want to show you in a few minutes how we can get really, really good results, okay? All right, let me just start Avizo. Okay, open data. All right, so this is the raw data. I'm just gonna show you with the slice going from top to bottom. Okay, let me zoom in a little bit. So this is let's say a membrane material. I'm not an expert in the field, but in general, these guys, they create membranes, right? And then they want to measure these pores inside. I'm not sure if it's clear to you, but they're these circular regions where supposedly chemical reactions happen, right? So this is what we want to segment. Now, if you just use, let's say, thresholding, so I'm gonna try that, it's gonna be a mess, right? Because, I mean, as you can see, here I'm capturing the pores nicely, but a lot of the outside stuff is getting included as well, right? And so for me, I would look at this and say, well, for me as a human, it's sort of easy to recognize the pores, right? There's a clear structure to it. So maybe it makes sense to use the AI segmentation tool for this, okay? And that's what we're gonna do. And I'll show you how to do that in less than, well, okay, I'm not gonna say less than 5 minutes because I'm gonna talk about it, but it's gonna be really, really easy. So again, we're gonna click the new segmentation workroom. I'm just gonna check this now just so we don't get this prompt all the time. So again, we're gonna specify the input data, and then it loads it here. And then, because we don't have any output label yet, I'm gonna create a new label, and this is my output. Optionally, you can just rename the material, just gonna call it pore. And now, I'm gonna create a patch set, okay? So this is something we haven't seen before. So I'm gonna create a patch set, and I'm gonna specify the size. So you have options here, which are multiples of 32. And I won't give much detail here, but the patch set is basically kind of the size of the input image to your deep learning model, okay, to your neural network. And for this example, I'm gonna choose 64. So this is one of the parameters you can play with when you're dealing with your own data set. And if you remember, the first step, or let's say the next step, is to provide the model with examples. And that's what we're gonna do. So I'm gonna click over here, and I'm just gonna click this. So I click this one here so we can focus on this window. And I'm gonna select a region. So I'm gonna do some manual brushing here to create some samples. So I click the brush button here on top, and I'm not sure, yeah. Hopefully you can see kind of the circle, that's the size of my brush. It's too big. So I'm just gonna adjust the size. Because I'm lazy, I'm setting it to the size that's exactly the same as these pores. And just look at this. So I'm now doing the manual segmentation, right? This is the human input. And I'm not gonna do all of this slice, but just a few. And I'm not an expert here, so I'm just using my judgment. And then, so I did the selection as usual, right? So you see the red highlights. And now I'm gonna click pore because I wanna add them to pore, and just click add, okay? All right, and now they turned green. Let's change that color because it's not very obvious. All right, so this is my input, right? This is the training data that I'm gonna give to the model. But before I can use, so actually this is the AI segmentation tool, right? That's the icon. So if you click that, you're gonna see on its window that it says this tool requests some patches. And we already created a patch set, but it's empty. So what we have to do is click on this button over here. So when you click that, you're gonna see this red square appear. So this is basically kind of a stamp. So you're gonna stamp on different regions in the data. So I click over there. It's not super clear, but it's adding these square regions in the data. And what you want to do here is basically create stamps around the region where you put your segmentation. And the algorithm, the training algorithm, will only use these regions, right? And okay, just to be sure I get some nice results, these are inside the squares as well. I'm just gonna make sure I segment those. So, all right, moment of truth. Let's try to run the segmentation tool. There's a few parameters here that you can change, but you can keep it for the first run. And then just click Create Selection. And yeah, so the training itself, it uses your computer's GPU. And depending on the size of your data, this can take a while, okay? Yeah. Yeah, that's fine. No, so it's fine. I mean, it's okay if they overlap, but basically what it's gonna do is it's gonna treat, it's gonna pull out those patches and treat them as like different images. Because under the hood, it doesn't have to know that they're overlapping, right? It can just think that, oh, these are different images. But yeah, I mean, you'll see that in the next example later. It's good to take, I mean, it's required actually to take patches from regions without the object that you're trying to segment, because it needs to know that, oh, in these regions you don't find these objects, so I'm not going to detect them there. Exactly. Yeah. So the more, the better, of course, but the more time you'll have to spend. And with this particular example, because the data is quite homogeneous, right? Like the regions with the pores are all the same, and the region with the background, it's all the same. I only took kind of two regions, right? There's a tricky region, which is the boundary. So I also tried to take examples there. But yeah, the more you use this, the more experience you're going to gain. And as you can see here, like the red regions are the predicted parts, which is pretty good. I mean, let me zoom out, right? And what's amazing here is that it did not select anything outside, right? And if you looked at the thresholding before, because it doesn't care about like where the data is, it was just selecting everything. With this one, at least it's getting just samples inside. And now if, again, this is just highlighted, it's not yet included in your segmentation, so I will just add it. Oops. I will add it. And now if I just go back to my view here and see it in 3D, it's actually there. And if we go outside, I just want to show you. So it's this one. Again, I will use voxelized rendering. You can see actually that it's getting these spherical, yeah. You see? I mean, that's really amazing. Actually, in the paper that they submitted, they just used thresholding, so the results weren't as good as this. But yeah, now you have that option. It was just a few minutes, right? I mean, look at that. It's really getting the nice shapes, but it's not perfect, right? So let me show you. Again, let's do the slice, color wash. So for example, in these regions over here, because the data is a bit noisy, you see, right? Like even for us humans, it's difficult to find the pores over here. And it's the same for the algorithm. So what you'll probably have to do is, if you can, or if you want to, you'll have to do some manual annotations here and do the training more. Yeah? Ah, yeah. So the question was just to have to zoom in and see how many pixels. Yeah. Yeah. Oh, yeah, sure. Yeah. You see? 1, 2, 3, 4, 5, 6, 7. Yeah, around. Yeah. Do you have a question about it, or? Yeah. Yeah, yeah. Yeah, of course, I mean, it's going to vary. For this data set, I don't know, actually. It's nano. This is nano. Yeah. These guys work on the nano scale. But that's pretty awesome. I mean, this is the first data set actually where I used the AI segmentation tool, and to me it was amazing. Of course, it's not perfect. In fact, I mean, if there was time, I was going to show.
I also would like to comment on this image. Diffraction patterns.
I have no idea. What do you mean, diffraction?
I don't know how it works, but object there was happened from the way.
Uh-huh. Yeah. Yeah, I mean, it depends on what you're imaging. Yeah, so I mean, it depends what your imaging device captures, right? And ultimately, you're dealing with the numbers, right? From 0 to 55 or 0 to whatever your is that an alarm on this building or? Next one? Okay, let's assume it's the next one. Can you check, Tom, please? Yeah, just to make sure we're not going to burn. All right, so I hope everyone agrees that for some cases, this is actually pretty cool, okay? And you are going to work on the next one if you want. But I wanted to show this. So this is the sand data set that I was using in the very first example. And this one we've seen that clearly, at least for me, thresholding works really well, right? Because it's just super easy to separate the sand from the air using the intensity values. But I tried using the AI segmentation tool on it, and it doesn't work right away, right? I mean, maybe it's gonna work eventually, but you'll have to tweak it more, okay? So it's maybe more tricky. So the main message here is that if you have a, let's say, simpler data set where it makes sense to just use simpler tools, just use those, right? Yeah.
Sorry, maybe you're right. It was not. About your why AI did not work, because maybe size of one grain was.
Maybe. So I didn't play with the parameters. I just used the default parameters.
From image I see like 50 per grain. What's your
No, this one, like you can see the patches here.
I mean 50 pixels per grain. But if you compare the
Yeah.
size of a rectangle, it can
Yeah.
all single grain size.
Exactly. Yeah. Exactly. No, that's a good observation. So remember I said like one of the parameters you can tweak is the size of this patch, right? And that's correct actually. So ideally you have to set the size of the patch so that the whole structure fits in the patch, right? So that the model can learn this. I don't have much experience in trying bigger patch sizes, but yeah, I mean, if you just think about it logically, it should. I mean, if the depth is the same, right, and then you're increasing the width and height of the image, computation should go higher. Yeah. But in general, I just played with the size 64 and 96. Yeah, something's wrong with these displays. It's flickering. All right, so now I'm gonna. Is it our building? Okay. Okay, yeah, okay, that's good. All right, so who among you has the deep learning environment installed? Okay, that's fine. So we're just gonna go through this example, right? So when you get access to a machine with a GPU and the deep learning environment, if you want to try this example, yeah. No, unfortunately, the AI tools only work on Windows. So this is a main limitation. We're not sure yet if Thermo Fisher will eventually support it. But yeah, for now it's Windows. For Ilastik, you can use it later. But yeah, unfortunately for the AI tools, only Windows. Yeah, and you need a GPU. So these laptops are most likely not gonna work. Okay. So for now, I'm just gonna show you where the files are once you have access to a machine with the GPU and Windows. You can try it out. If you don't have, we have a machine in the Vis Lab that you can book, that you can use for these kinds of tasks. Okay, I'll show the link later on. Huh?
How to book the machine?
Yeah, I'll show it it's in the last slide. But if you go to I can show it now. If you go to wiki.vis.kaust.edu.sa, that's the KVL website, go to facilities, you're gonna see the calendar of the availability, okay? And then on the bottom here, if you log in using your KAUST credentials, you're gonna see the facilities you can book. And it's called the Vive, okay? It's called Vive because we use it for VR, mainly for VR, but because it has like a nice GPU, lots of memory, some students book it for using Avizo, okay? If they don't have a powerful workstation. Okay, anyway, going back to this example, so this is actually an example from Avizo, from Thermo Fisher, right? So I will share the slides later on, so you can visit this page where the files are available. But I also shared the files in the datasets folder. If you go to cryo-EM folder, and it's super nice because they actually provide a video that you can follow step by step, okay, on how to do this. Okay, I'm just gonna skip it. So you see, it's the same process, right? And yeah, you can watch it in your own time. But for today, I'll just open the pre-configured project, right? So this is always nice because everything is there already. So we'll save some time. Amira Avizo is the same. So yeah, it's the same conceptually. It's just different name. It's more of a historical thing, right, Tom? Like they used to be different software, but they got bought and they got merged. It's the same software now. So yeah, if you open that project, you're gonna see this. So this is already the final segmentation. And I'll just quickly go through let's say the training input, so you have a feeling of how that looks like. And in this particular example, you have, I think, 4 different objects or materials. So you have these kind of fibrous materials or like microtubules. You have the pink ones, which are ribosomes. The yellow ones are the mitochondria boundaries. And these bluish ones are, they call vesicles, okay? But this is a result of using the AI segmentation tool. And let me turn off the 3D result. This is the raw data, right? Let me adjust the contrast. So this is the image that I showed earlier, okay? And let's try actually. Let's try do we have time? Yeah, we have time. I'm gonna go to segmentation. Why is this showing again? Okay, anyway. So again, we start off by specifying the input, and it's just this grayscale thing. So, okay, let's try. I'm gonna create a new label and a new patch set. Okay, 64. And let's try to segment the tubes. Right, so let's zoom in over here. Okay, so let me just brush. Let me zoom in some more. Okay, can you see it? I hope you can see it, but I'm gonna brush on these tubes. I've never tried this before, so we'll see how good it will be. Anyway, we'll do a quick try, and then if it fails, I'll show you the good results. And obviously, the better your manual segmentation is, the better the results of the AI segmentation. And it's obviously difficult with the mouse, right? And to help with this, we actually have a pen and tablet interface in the VisLab as well. And you can book that when you book the Vive machine. It's attached to it. And I'll show a video of that later. So anyway, let's try this. Add segmentation, patch. So let me just click here, and then I'm going to click on regions without it. All right, now let's see. Create. So while this is computing, I just want to talk a little bit about the parameters you can set. So here you have the number of epochs. So this is for the training, right? So how many iterations the training will happen. And you have this elastic deformation strength. And that's basically, if you're familiar with data augmentation, this is kind of the higher this number, the more data augmentation happens. So what it does is it takes your input patches and it kind of stretches them a little bit, rotates them a little bit, just to introduce some variation to the training data, right? So it artificially increases your training data. So this is the result, and it's not super good. I was kind of expecting that. Yeah, so it's missing a lot of stuff, but I think my annotation is pretty bad. But you get the idea, right? It's how do I say it? Obviously, the better quality of your training data, the more chances of getting good results. So you have to spend more time on it. Sometimes maybe you have to tweak the parameters. So there's a bit of an art into it and a bit of trial and error, right? And for most of you who have deep learning experience, it's just that, right? I mean, it's just a lot of trial and error, okay? So I don't want you to be disappointed, so I'll show the better results. So let's look here. Let's look at the tubules. Okay. Just to give you an idea, let me clear. All right. So what you see here is, so the green is the ground truth that's provided in the project file. And the blue regions here are the patches. And I'm pretty sure there's patches in other slices. There, yeah. So you can add patches from different slices, okay? So I forgot to mention this. So of course, for instance, in some data sets, like there's some slight variations in the grayscale from the top to the bottom. So it's good to, I mean, of course it's best if you have a consistent grayscale, but if that's not the case, you can take patches from different slices as well. And let's take a look at the final result. Oh, actually, this is already the final result, the green part. And yeah, so these green parts inside the patches are the input for training, okay? All right, so I won't go through all the materials, but you can take a look here, okay, to get a better feeling of how to do it for different materials, okay? Yeah, so that's let's see. Yeah, so we're gonna take a break, and after that, we're gonna switch into Ilastik. So before you go for a break, just to make sure that you return. So the nice thing with or a few words about Avizo. So Avizo is a commercial product. Okay, so KAUST has a license. So while you're inside KAUST, inside the KAUST network, you can use it. And as I said, the AI tools only work for Windows right now, and you need a GPU. Okay. Now Ilastik is an alternative, right, for segmentation. It's cross-platform, so it works on Linux and Windows, maybe Mac OS as well. It's free and open source, so it's free, which is amazing. What else? And you don't need a GPU. So actually for today's demo, I'm just going to use CPUs, okay? So you have the option to use CPUs or GPUs. Of course, if you have GPUs, it's going to be faster, okay? All right, so let's take a 15-minute break. Yeah, I think we'll just continue. All right, okay. So, yeah, now we'll move on to Ilastik, which is an alternative, right? It's doing similar things as Avizo, but it's more focused on segmentation. So it doesn't have the analysis tools like Avizo, right? Like counting the particles, measuring, etc. It's focused purely on segmentation, okay? But I'll show you in a little bit that you can export the results of Ilastik and bring them into Avizo so you can do your analysis in Avizo. Yeah, so as I mentioned before, the advantages of Ilastik is that it's cross-platform. You can use either CPU only or GPU, and it's free. And the installation instructions, or actually just the link to the installer is in the training webpage. All right, so does anyone have Ilastik installed? And we'll do the hands-on. All right, cool. So I'll just go through it, and then you can try it out later on once you have it on your machine. So I'm just gonna run it right now. Okay, so I'm using the latest version of Ilastik, which is 1.4.0. And so in the beginning, this is kind of like what you see when you open the application, and you have different workflows here. And what we will work with today is this neural network classification, right? And from the name itself, it uses neural networks, so deep learning for doing classification. Later on, I'm also going to show pixel classification, which is, I mean, for the past few years, this has been the workflow that we've been teaching people, right? Because it still works, and it has advantages over the neural network classification. And I'll talk about that later. Okay, so for now, let's do the neural network classification. So I just click this. And just a quick thing, so you see here you have two options for neural network classification. We have local and remote. And from the name itself, local, it means you're gonna run the computations locally on your machine. Remote means the computations will happen on a different computer, right? So if you have, let's say, bigger data sets or your laptop doesn't have a GPU, you need more compute power, you can use a different machine. Okay, like IBEX, for example, here in KAUST. So for now, I'll just use local. And then you just, in the beginning, you have to specify a project file. So I'll just use the default and put it in some folder that you will have access to later on. So this is the project file that will save your files inside, your settings, etc. So you can come back to it later on. And this is Ilastik. It's very minimalistic, which is nice. And on the left, you kind of have this panel that has these numbered steps. So this is what I like about Ilastik. It's a clear step 1, step 2, step 3, step 4, and so on. So right now we're on step 1, which is you specify the input data. So here on the right side, I'm gonna click this add new button, and I'm gonna say add separate images. And in the datasets folder that I shared with you, there's these mitochondria images. So I'm just, for now, okay, let's load the multiple slices, the mitochondria data set. All right, so once you're loaded, the image slices show up here. So you can use the mouse wheel to scroll through the slices. And for this data set, we want to segment the mitochondria and the boundaries, okay? And we're done with step 1, right? So that's specifying the input data, and now we go to step 2, which is the neural network prediction. And here is the interesting part, right? So in Avizo, we just had the deep learning model built into the application, right? So you don't really have a choice on the type of network. You only have a choice on specifying the training data, okay, and a few parameters. In Ilastik, the neural network classification, right now, you cannot really give it training data, right? That's a bit weird because all you can do is select a pre-trained model from a collection of models. So if you go here, there's a download models button. If you click that, it will bring you to this bioimage model zoo. And basically, what this has is a bunch of pre-trained models that you can try on your data, okay? And I mean, it's nice in the very front, you have these pictures that kind of give you a preview of what the model can do. So for instance, this one looks like something that we can use, right? So it's segmenting the mitochondria plus the boundaries. This one, for example, is for confocal, segmenting the cells, individual cells. This one, another for confocal, for segmenting just the nuclei. So you can click on the model. You can take a look at some documentation about it, citation. Sometimes they give you links to the paper that describes the model. And let's say, right now there's not a lot. So these are all the models that are out there, but this is pretty new, so hopefully in the near future it's gonna keep increasing. But this is super low effort stuff, right? Because you don't really have to do anything except copy the model. So to copy the model, you just click on the model. And you see here, there's two things, right? One is the kind of a DOI, like a unique identifier, and some unique name. So you can just copy this, and that's all you need. You go back to Ilastik and type, or in this case, paste the name of the model. Right here, shivering raccoon is just some random animal names. And then click this to activate the model, okay? And as you can see, it will download the model, right? It will download the model and set it up for us. But for this new version of Ilastik, or I mean, right now there's an issue, so it crashes. And I mean, I'm in contact with the Ilastik team to deal with this. So for now, the way around this is to set up a remote server on my local machine, right? And I'll do that right now. But for you guys, I put the steps over here. So if you go to the training website, there's a link here that says how to set up TikTorch on Ilastik. So the name of the segmentation server is TikTorch, for like combination of Ilastik and PyTorch. So TikTorch. So if you go here, there's the instructions here, how to bypass this issue with Ilastik. But hopefully in the next iteration, this is going to be solved, okay? So I won't go through this. So I already did the steps. And I will run now the TikTorch server on my machine. So you don't have to do this later on once the issue is fixed. So basically, I'm running a compute server on my laptop. So if you want to do the remote workflow, like let's say on IBEX, you'll have to do this on IBEX, right? Install the server there. And now if I run Ilastik again, now I'm going to choose the remote option, right? Again, once this issue is fixed, all you have to do is the local. But for now, I'll do the remote, start a project file. And now there's just an additional step in the beginning, which is the server configuration. Otherwise, everything else is the same. And I just check basically the IP. Yeah, so, or the port is 5567, and because it's my own machine, it's just 127001. Get devices, it's going to show you the available devices on that server. So right now, only the CPUs are set up. And that's it. Okay, it was just telling Ilastik all the computations will happen on this machine. And it's now the same steps, right? So again, I'm just going to load the data. The same data set. And then the shivering raccoon. Yeah, so without the issue, you will get to this step, and then this button will be enabled, which is the live prediction. If you click that, it will then use the model to do the prediction on your data set, right? So try to find the mitochondria in the boundaries. So it's kind of progressive, right? So it takes some time to do the computation. So for example, for this slice, I already see the preview. So the yellow is the mitochondria and the blue is the boundary. So I'm just scrolling through the slices, and as you can see, I'm getting results. Yeah, so it's not perfect, right? Like you'll see here, it's not perfect, so you'll probably have to do some fixing. But it's basically free, right? You didn't really do anything. And if you're lucky, it will just work perfectly on your data, right? So this is the disadvantage right now. You can't really tweak the model. So it's, yeah. So if you want, you can then, if you're not happy with the results, you go back here and pick a different model and then try it out, and so on, okay? And now, I mean, it's not perfect, but let's say I'm happy with the results and I'm willing to do, let's say, manual corrections. Let me show you now how to export the results so that I can bring them, for example, into Avizo. So if you go to the next step, data export. Yeah, now we go to choose export settings. These are the different channels that you can export. So Z is the stack, YX is just the width and height. C is the channel. And this is a bit tricky because we have two materials, right? So the mitochondria and the boundary. If you export them at the same time into an image, the output is weird. So Avizo is not able to interpret them properly. So you'll have to export the channels separately. So for now, I'll export the first channel. And then I'm going to specify the format as a TIFF sequence. And then I'm just going to keep them in the same folder as the data and click OK. And then click Export All. And now if you go to the folder with the data, oops, yeah, it's going to create these images with the segmentation, right? And as you can see here, it says probabilities, because the output for the neural network classification is a probability that a pixel belongs to a class, okay? I'll show you in Avizo how that looks like. So let's start Avizo. Now I'm going to open data. Okay, let me All right. So this is how it looks like. And if you look at the data object, it's actually floating point values, right, of probabilities. And if you want to get segmentation, like integer values, you can use, for example, interactive thresholding, right? So you can say what probability you're willing to accept, right? So this is up to you. So yeah, I'll just set something and then click apply, and out comes your segmentation data, right? And this is now your mitochondria, right? I mean, it's not perfect, as I said, so you can do manual fixing if you want. But then you can do your further analysis here, right? Like label analysis. If you label analysis, so if you want to count the number of mitochondria, for example, measure the surface area, volume, etc., you can do that in Avizo, okay? All right. So that's kind of how it looks like. Super easy, actually, right? You just give your input data, pick a model, load the model, apply the prediction, export to Avizo, okay? As I said, the disadvantage is you don't really have control over the model, okay? That's pros and cons, depends how you look at it, because you don't have to really build anything. All right, so now, yeah, I want to show the pixel classification workflow as well. So this is not using neural networks, it's using random forest. It's still in Ilastik, and it's quite useful. So I just show it quickly because I think this is a very powerful tool. So for me personally, I first try segmentation in Avizo. If that fails, I use this, okay? So let me load. I've in the datasets folder that I shared with you, if you go to Ilastik folder, there's a Ilastik folder. There's this cell classification project that I already set up. So this is a confocal microscopy example. Yeah, and this dataset is from Stephen from the Imaging Core Labs. And this is how it looks like, right? So let me just, yeah. So for this data set, I want to extract these like tubular structures from the cell. And if you use thresholding in Avizo, it's not gonna work. So here I provide, sorry, I should have shown the steps, but I'll do that in a little bit. But here, just like the AI assisted segmentation in Avizo, you have to do some brush strokes, right? To give it examples. So here, for example, I can brush manually, right? Oops. You get the idea, right? So I have the brush, and let's say this is background, this is what I don't want, and the yellow are the tubular structures that I want. And once you're done with that, you can click Live Update, and then it will show you the preview of the automatic segmentation, right? You'll see it here. So here you still give some training data, but instead of a deep neural network, it's using random forest. And then once you're happy with the prediction, just like before, you go to export. And in this case, you have the option to export segmentation already. So it's going to do the thresholding for you. So let's try that. And because there's only one channel, we don't have to change this. And let's say I do PNG sequence. Let's see how it looks like, and export. I'm going to export again into Avizo. Yeah. So now it's going to generate the. So while this is computing, so this is very similar to, well, The difference here is that you provide the random forest algorithm the input for training. Okay, so you have more freedom and more control, but it requires more effort, of course. Okay, so this might take a while, so I'm just going to load whatever's there. So let's start a new project. Okay, so I'm going to go here, just load whatever is done. Okay, so yeah, so the first thing you'll notice when you look at the data is that it's just black and white stuff. That's because if you look at the loaded data object, it treats the data as an 8-bit unsigned grayscale data. So first we'll have to convert it. So convert image type to a label field, which is a segmentation data. And so clean labels, let's try. This usually subtracts 1 to the data so that everything starts at 0, but I don't want to do that. All right, so if you do that now, you get this, right? So the dark blue is the background, and the light blue is the segmented tubular structures, okay? And yeah, so if you want to just see the tubular structures, let's do arithmetic. So the arithmetic module just performs an arithmetic operation on your data. So in this case, I will remove the ones and set them to 0, so it becomes background, and set the 2s and set them to 1. Actually, let's do a logical operation. A bit confusing, but what this does is that it's saying, okay, everything in the data object that's equal to 2, set that to 1, right? So this performs a logical operation, otherwise set it to 0. Okay, and what that gives us, oops, actually it should be 1. Yeah, and then what that leaves us is just this. And then everything else is 0, and now we can see this in 3D. Yeah, so now you have your 3D structure, okay? All right, let's see. Oh, actually, so this was that example. And now, yeah, so you can also take a look at Ilastik and see some of the models that deal with this type of data. Okay, so I think for now that's it for segmentation. And we've seen how to use Avizo's basic tools for segmentation, the AI tool for segmentation, Ilastik's neural network classification, and Ilastik's pixel classification. And let's say now you have your segmentation data, what can you do with it, right? Because in the end, you want something that you can put in your paper, right? So there's a question you want answered, and the segmentation is usually just a means. What you want to do in the end is to apply some measurement or analysis, okay? And I listed here some of the common analysis and measurement modules in Avizo, so you can take a look later on, but we'll go through some of them today. And the other tip is in Avizo, if you have your data, if you just click over here in this circle and look for measure and analyze, it will show you the modules that you can use for analysis, okay? All right, so I'm just going to go through 2 data sets today to show you some of the common measurements and analysis tools. So the first one is the CT of a coral. So let's go ahead and do that. All right, so I'm going to open the data set. I shared it with you as well. Coral, okay. And as always, I would inspect the data first. So usually using the ortho slice, but even better with the volume rendering. So you see kind of the 3D structure of the data, right? So this is how it looks like. And if you look at the slice, so this allows you to see inside the data, you're gonna notice like there's kind of pores inside, right? So I'm not an expert here, but for now I'm just gonna call those like pores or like less dense materials, and there's the dense region. And for today, I'm gonna show you how to measure the dense material and the non-dense material and see the fraction or the percentage. So the first thing I'm gonna do is use thresholding, right? So once you attach the thresholding, it actually gives you a recommendation on the range to use for segmentation. And here, let's say I'm happy with this, and I'll just hit apply. And now I will get I got the segmentation already of the dense material. And the next thing I want to do is to get the low density material inside. And one technique to do that is, you can use thresholding. Let's actually try that. Let's turn this off. So you can do thresholding, but adjust now the threshold to the lower intensity values, right, to get the stuff inside. But this is a bit tricky because the background also has low intensity. So you have the risk of capturing the outside as well. So in Avizo, there's a tool that you can use to only segment what's inside this already pre-segmented material, and that's called ambient occlusion, compute ambient occlusion. So let me just run this. So conceptually, what's happening is that inside each pixel, it's gonna shoot arrows going out, and it's gonna count how many arrows hit an object, right? So if you imagine if you shoot an arrow inside here, all around it, it's gonna hit an object, so it's gonna count many hits. And for outside, if you shoot arrows, it's only gonna count a few. So if you look at the result, it actually looks like this. This is the result of the ambient occlusion. Okay, and then you're gonna want to apply a thresholding on this one, right? So you just say, okay, I want to include in my segmentation those regions where I hit many things around it, right? And then hit apply. Okay, so now I have a segmentation of the dense material and the low density material. And let's say I want to combine them. So I'm gonna do arithmetic operation. Okay. So what I'm gonna do is, so A is the first material, the dense materials, and I'm gonna say that's has a value of 1. And then the low density material, in this object, they also have a value of 1, right? But if I want to combine them together, I'm gonna do A plus B times 2. Okay, so the A will still be, sorry, it should be like this, yeah. So, okay, no, the result. Yes, okay. So now you have both segmentations inside, right? So the low density and the high density are together. And now you can do volume fraction. So volume fraction from the name itself, if you click apply, so it will count the number of voxels that belong to the two materials, right? So you see here a table, I'm not sure if it's clear, but you get the number of voxels for the first material and the number of voxels for the second material. And using that, you can compute the percentage, right? So you can say there's X% of the dense material and Y% of the low density material, okay? All right, and the last one that I want to show for this coral is the measurement, because someone said they want to measure the length of some objects. So for example, for this coral, you want to measure the length of a branch. Let's say this branch you want to measure. So in Avizo, there's actually these measuring tools over here on the top. Okay, it's not super clear, but there's like an icon of a ruler. So if you click that and click inside, so you can start, click on the starting point and the end point, then you can get the length. But this is not always accurate. So you see here, it's not always aligned. So the best thing to do is to create a slice over the part that you want to measure. And to do that, we're going to use a slice module. Okay? And it starts off as orthogonal to the data. But, okay, let me just move it close to the object I want to measure. And then I'm going to clip the data. And now if you click this rotate checkbox over here, it's going to show you this kind of control, let's say, icon that allows you to reorient the slice. So you can orient the slice perpendicular to the branch that you want to measure. And now, if you click the ruler icon again for measurement and click this accurate, you can click on the tip and into the core. And this now is a more accurate measurement. So as you can see, it's perfectly aligned. It's perfectly aligned to the branch, so you get a more accurate measurement. And the measurement is here, so it's 1.7 microns, okay? All right, so that's one. And now finally, we'll go to the last data set. So I'm just gonna close this. So for the sake of time, I have a preloaded project here in the datasets folder. So I'll just load that. Okay. So this is a core plug sample, right? So for people in Antwerp, this is a pretty common type of data where they have rock samples or soil or some geological sample, and they have a core sample of it, and they do a CT scan, right? So they do a scan, and a very common analysis for them is pore space analysis, right? So they want to see the pores inside the rocks, because the pores is where the fluids flow, right? Like oil and water and gas, for example, and they're interested in this type of flow, okay? And while this is loading, just to give you a preview of what analysis we did here. So usually you would do a filter to denoise the data, and then usually thresholding is fine to extract the pores. So let me show the steps one by one. So we start off with the raw data that looks like this. So it's just grayscale value, right? And you can see the pores already in there. And if I do a volume rendering, it's even, so you see it here better. So you can already, in this particular example, you can clearly see the pores, right? And this is what we want to extract, right? And here actually, even with just interactive thresholding, it's already enough to extract the pores, okay? So we do that, and then we use the separate objects module to separate the different pores from each other. Yeah, so the result looks like this. And then we use the label analysis module to assign, a different ID for each pore, and it looks like this. And then you have a filter by measure module. And this one, if you look here, you can filter the data based on different measurements. For example, the volume, surface area, etc. So for instance, you're just interested in seeing the biggest pores. So you apply the filter by measure module, and then you get a result that looks like this. So this is just showing you the biggest modules, okay? And for example, if you just want to see the smallest pores, you do the opposite. And yeah, so this is a very, very common workflow. So once you do the segmentation, you separate the particles, and then you do label analysis to get the statistics. So by the way, these statistics of the 3D volume, the surface area, etc., you can export this into an Excel sheet, for example. So if you just click here, export, you can export it into CSV, which is comma-separated values, and you can open that in a text editor or Excel or Google Sheets, okay? All right, that's that. So that's the last example. So there's more measurements and analysis modules in Avizo, so I encourage you to just explore what's out there. There's a user guide over here, so if you click help, user guide, you just search for a keyword here. Usually you get something meaningful. But yeah, there's a lot of modules out there that you can try and use. So for me, this is kind of my personal workflow recommendation when I'm dealing with a new data set. So first of all, I inspect the data in Avizo. So use the orthoslice, volume rendering, you check the histogram, get a feeling of how the data looks like. Try, if you have more experience, you get a feeling of what kind of tools will work. If not, yeah, try to follow this workflow. So if the data is noisy, try to filter the data to reduce the noise. Some popular filters are median filter, bilateral filter, non-local means filter. If you need help trying to figure out what the best filter is for your data, you can come to us, and we can suggest some. After filtering, try the simple segmentation tools in Avizo, right? So thresholding, watershed, texture classification. As I said, we've done workshops on these tools before, so just look in our YouTube channel, search for KAUST Visualization Core Labs in YouTube, and you'll find our channel with our previous workshops. If that fails, then you can try the AI Assisted Segmentation in Avizo. Try to tweak the parameters. If that doesn't work, try the segmentation tool in Ilastik, okay? If that doesn't work, try the pixel classification in Ilastik. If that doesn't work, there's actually a more complex deep learning segmentation tool in Avizo. So for this one, you actually have more freedom in terms of the parameters you can tweak. So you can tweak the learning rate, the type of network, the optimization algorithm, etc. So I didn't show it here, but if you're interested in trying it out, let us know or reach out to us. There's other tools out there. I'm going to show some of them in a little bit. Worst case is manual segmentation, right? But this is what you want to avoid. But I had a student who actually did manual segmentation. So she worked on her data set for 1 week, and she got perfect results, right? Because this is kind of the best you can do, but it takes the most time. And to help with that, we actually have this pen and tablet interface, which I'll show later. So this could help speed up your segmentation. Instead of using a mouse, this can speed up your process. If you're not sure, you can reach out to us and we can help you come up with a workflow to improve your segmentation. So as I said, there's a deep learning training and prediction module in Avizo. If you're interested in trying it out, it's called DL training and deep learning prediction. So obviously DL training is the training part. So you give it your training data, as you see here on the right. And then you specify the parameters for training. There's a nice tutorial in the user guide in Avizo. So if you just search for deep learning, you will find this. It's a step-by-step, so you can follow that. And once you're done with the training, you use the deep learning prediction module to load the model you trained and then use it for prediction, okay? And there's other tools. So Fiji, I think this is a tool that's been being used by the Imaging Core Lab. So there's plugins for Fiji, like this one that you can use for segmentation. MATLAB, OpenCV, these are more like high-level tools that also have segmentation tools. If you want to go lower level, so this is more, I would say, more involved process. You can use TensorFlow and PyTorch. So there's models out there, like when people publish segmentation algorithms, sometimes they would publish the TensorFlow and PyTorch models. Then you can just load those and use them, or you can tweak them as well. But this would involve you really dealing with the Python code and like really tweaking the models, okay, and playing around with those. And finally, as I said, you can do manual segmentation. And this is a video showing the tablet and the pen and tablet interface that we have. So it allows you to like more quickly do your manual segmentation, right? So you can do the brushing. There's a lasso tool that you just like specify the outline and it will shade everything inside. And you can book it through this link, use your KAUST credentials. And it's not only the pen and tablet interface, but the machine itself has, I don't know, like 700 plus gigabytes of RAM. It has a nice RTX GPU that you can use for deep learning segmentation. And in general, some students use it for like heavy lifting of, or I mean, heavy computations with Avizo, okay? And that's it. So again, if you want to know more about the Viz Lab, you can visit our wiki, and if you need more specific help, reach out to us at this email. And for those who are here, I would appreciate it if you can provide feedback in this feedback form. It's just 2 clicks. And yeah, if you can share a few words, both positive and negative, some feedback to help us improve the future workshops. Yeah, so thank you for coming, and reach out to us if you need any further help. Okay, thank you.